Okay, so now this is part 2 of my description of this supplemental gene regulation PowerPoint. And so this is again that model that describes all of sort of what we know about transcriptional regulation. I mean, summarizes it. It's not everything we know, of course. So we covered a lot of stuff about cis-acting sequences, proteins that bind them, DNA binding domains, activation domains. We got our megablob, we got our general transcription factors, et cetera, et cetera. What this part is about is how we go from condensed chromatin to loose chromatin, and how we go from loose chromatin to condensed chromatin, and some of the experiments that we use to be able to tell that. Okay. So, now even though this conceptually, at least in my mind, sort of happens first, that you have to loosen up the chromatin before you can bind DNA and stuff, this other stuff was discovered first because it was just easier at the time to sort of work with purified DNA and purified proteins rather than chromatin. So this is what we've done so far. So we've done all this part already. So we talked about some of the experiments that we use to identify cis-acting sequences and transacting factors. And now we're going to talk about this part. And we're going to talk about some specific histone modifications and how we go sort of back and forth between condensed and decondensed chromatin and how that affects gene expression and the experiments that we use to figure this stuff out. So as we know, transcription also involves changes in chromatin structure. And there are various kinds of chromatin structure where we can sort of abstractly just kind of represent it like this. Here the gene is off, because it's scrunched up and the proteins can't get to it. So you've got your transcription factors out here that are trying to get to the DNA, but the DNA is scrunched up and hidden in that condensed chromatin structure. So this is condensed. And here the genes are on, because now that it's opened up, maybe these cis-acting sequences are available to be bound by proteins, and eventually you can turn the gene on. So that's sort of the abstract view of what it looks like, just kind of the overview. And then what are the kinds of things that can loosen up chromatin? We mentioned before that acetylation can do it. And what kinds of, and actually deacetylation, if you take those acetyl groups back off, that'll close up chromatin. Also methylation will close up chromatin, and demethylation will open up chromatin. And also DNA, so this is histone methylation, but you can also have DNA methylation that has a similar effect. And notice I keep saying chromatin, 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 chromatin. So it's not DNA. We're not talking about DNA. We're talking about chromatin. Chromatin has DNA and proteins. And the DNA is wrapped around these histones, and you've all described to me how a nucleosome consists of 8 histones, and the DNA is wrapped around 146 base pairs, et cetera, et cetera. So you have a vague notion, at least, of what a nucleosome looks like, and that there are histones that are involved. So that's what we're talking about when we talk about chromatin. And so the idea with these acetyl groups and methyl groups is that there's also a lot of other groups. And so here's just sort of a summary of different types of groups that are found on histones. And each of these groups will affect histone structure in different ways. And we're just going to talk very generally speaking that acetylation opens up chromatin and methylation closes chromatin. You can also have all these other things, phosphorylation, ubiquitination, and other types of things. For the most part, we're going to look at, and you can actually, people study each one of these different types of marks specifically. And let's see. So an important one is histone H3. So a lot of times you'll see something like histone H3, and let's just use an example, K9 acetylation. So that would be this. So this is number 9. There's a lysine at number 9, and it's acetylated. So we can talk specifically about H3K9 acetylation. There's H3K27 is a big one. So here's H3K27. Now I'm not gonna, you don't have to, at this point, memorize the specific modifications at the specific histones that cause these specific changes. Right now we're just talking about generally that acetylation loosens up chromatin and methylation closes it. But what we also see is that there's lots of possible marks. They call these marks, or they're chemical marks or modifications. And so if all of your histones have all these different marks, you can look at this as a layer of information that's on top of the DNA. So this leads to this idea. So we've talked about you've got a gene, and that you have the same gene, and it might be in the kidney, might be in the liver. This gene might be on, this gene might be off. Well, one of the reasons that this gene is off is because it's scrunched up in chromatin in these cells. And the reason it's on is it's loose in these cells. And so we can transcribe it. And the reason that this is tight is maybe because it has methyl groups on it. And the reason that this is loose is maybe because it has acetyl groups on it. So even though we still have the same DNA, we have a different phenotype. The phenotype is, I am now a kidney cell. The phenotype here is, I am now a liver cell. And the difference is not the DNA. The DNA sequence is the same. But there's more information. There's more information contained in this histone modification system. And so there's a guy named David Alice, his name's C. David Alice, this guy I know from Syracuse. If I can find a picture of him, I'll show a picture of him. I mean a picture of me with him. It's easy to find a picture of him. He's a pretty famous scientist. Anyways, so he came up with this idea that there's this different layer of information on top of the DNA information. So here we've got our DNA information. And this is the same sequence, whatever it is. Exact same sequence, no mutations, no nothing, exact same sequence. But this has a different histone modification pattern than this, right? So they're different. So if we call this the genetic code, is the DNA code that we all know about. This level of information is called the histone code. And so David Alice and some other folks have come up with this idea of a histone code that is an extra layer of information on top of the genetic information. Because it's information on top of other information. So we got our genetic information. That's our genetic information. But on top of this, with our DNA all looped up into these nucleosomes, and these nucleosomes have different modifications on them, this extra layer of information is in addition to and it can mask or change how the genetic code is interpreted, and so we call that epigenetics. So that's on top of the histone, that's basically the histone code is one idea of of this epigenetics. That epigenetics is a layer of information that's coded for in histone modifications, and that's on top of or in addition to the information that's coded for in the DNA. very hot field, very important. It's kind of where things are at these days. All right, so how do we know? So that's always a good way to think about these things. Okay, I just told you that. How do we know? Well, we can have a hypothesis. Sure, it certainly makes sense that condensed chromatin would be associated with genes that are off, and loose chromatin would be associated with genes that are on. Well, how do we know? Well, let's take, so this is kind of a famous experiment. It took a gene, the globin gene, that's expressed at super high levels in erythroblasts. So erythroblasts are cells that become, we'll make them red blood cells. But all of the hemoglobin is made in these erythroblasts. And then when you have a red blood cell, you have all your hemoglobin. But the hemoglobin loses its nucleus. Or the red blood cell loses its nucleus. And so everything happens before the cell becomes a full-fledged, mature red blood cell. It's called an erythroblast. It's basically a stem cell that becomes a red blood cell. So it expresses a lot of hemoglobin, tons of hemoglobin. Globin, billions and billions of molecules. Maybe not billions, but certainly a whole lot. And this does not get expressed. So we know that going in. How do we know? Well, we could do a northern blot. We could take the take the RNA from erythroblasts and take the RNA from some other cells and do a northern blot, or do any other kind of experiment where we measure gene expression. And this type of cell, this is a different type of cell that doesn't express. So we just know that globin is not expressed. It doesn't matter what kind of there's pretty much every other cell, globin is not expressed. So this just happens to be some tissue culture cell that they use that does not express this gene. So they know the gene is not expressed. That's important. So now let's go in and see if chromatin structure correlates with this expression. And we would expect, this is our hypothesis basically, that when the gene is on, the chromatin is loose. When the gene is off, the chromatin is tight. So we're going to do an experiment. And this is this DNA's hypersensitivity. We've gone over it a little bit in chapter 17. This is different than the DNA's footprinting because we're looking at chromatin. We have to isolate chromatin from the cell. So the way this experiment works is isolate. What are we going to isolate from the cells? If we were doing a northern blot, we'd isolate RNA. If we were doing a southern blot, we'd isolate DNA. If we're doing a western blot, we'd isolate protein. But here we want to look at chromatin. So we're going to isolate chromatin. And chromatin is a specific thing. So we're going to isolate chromatin, and it will be either loose or tight, but it's going to be DNA with histones connected to it. Then we're going to treat the chromatin with DNase. We're only interested in a particular region. So now that may or may not chew up this chromatin. So if it's loose like this, the DNase will be able to get in there and chew it up and break this DNA down. It's actually going to break it down even further. Now we want to know, now we can ask the question, did the region near our gene of interest, was that chewed up or is that DNA still there? So now we have to isolate DNA. So we've already, so DNase is the key here, DNase, which cuts up DNA. We isolate DNA, and in this case, they do a southern blot. And a southern blot is a way to detect specific DNA. You could do PCR or something else, but they do a southern blot. And then you need a probe. So we're interested in this DNA right here. We want to know if that DNA was associated with loose chromatin or with tight chromatin. So what we'll see if we back up, when we do the DNA step, this DNA will get chewed up. Chewed up. This DNA will not get chewed up because it's protected by all that chromatin that it is contained in. So then we can do a southern blot where we take all the DNA and we cut it with some restriction enzyme. And then we're going to use a probe that hybridizes to the globin gene because we're really interested in just this gene. If we ran all the DNA on a gel and just looked at it, we'd have all kinds of DNA. We wouldn't know what we were looking at. So we want to use a probe that hybridizes specifically to this DNA so that we know if it's there or not. And what we predict is that the DNA is going to be gone here, but it'll still be here. Because if a region of DNA is sensitive to DNase, it gets chewed up and goes away. If it's not sensitive to DNase, it doesn't get chewed up and it stays there. And so when we do a southern blot, southern blots are like this. And you get a band if the DNA is there. And you don't, you get a lighter band if the DNA is getting chewed up. So here's the result of that experiment. So here's DNA. So they've isolated chromatin. So isolated chromatin, treat with DNase, do a southern blot, hybridize to a probe that sticks to the globin gene. So we call that a globin gene probe. Wherever we see a band, there's DNA is still there. So in these erythroblasts, where we suspect that the chromatin is going to be loose, that's our hypothesis at least, as we add more DNase, the DNA goes away. It's being chewed up. But in these other cells, where the gene is not expressed, we expect the chromatin to be tight. And here, this is our highest level of DNase. So here's telling us how much DNase we added. If we add 1.5 micrograms per mil of DNase, this DNA is pretty much gone. Don't worry about all those dots. That's just stuff. But here, the DNA is still present. So that tells us that this, when we originally isolated chromatin, this was tight and this was loose. When it was loose, it got chewed up and goes away. This is just a control. This is some other gene that they know. This is just to show that the southern blot worked. So that's just some other probe that they use for some other gene that they know should be there. And it's not the greatest southern blot in the world. You can see sometimes the DNA doesn't transfer over from the gel to the filter very well, and so sometimes you get little spots where you're missing stuff. All right. So that's an important experiment. That's called and so that tells us that that region that we use for the probe is a DNase sensitive site. They might call it a hypersensitive site. So we talked about DNase hypersensitive sites earlier. This is how you identify DNase hypersensitive sites. Okay, so that's basically it. And I'm going to reiterate this one more time. So the idea is, now we've got the same gene, different cells. Erythroblasts in this case. So instead of kidney and liver, we're going with the erythroblast. And we're going with MSB cells. Now what we know is that the gene is transcribed at a high level. Because if we did a northern blot, that would tell us, or qPCR or something would tell us that we get a lot of transcription in the erythroblast, no transcription in these cells. So now what we're going to do is we're going to do an experiment. We're going to do DNA sensitivity. And we're going to show that when we do the DNA sensitivity experiment, we take our erythroblast cells and our MSB cells, the erythroblast, the globin gene goes away in the erythroblast cells, but it's still present here. Now let me say that, be careful. It's not that the gene is not present. So the gene is always the same. The gene's present in both. But the DNA goes away after we treat it with DNase. Right? So this is the result of, so this makes that a sensitive and this a resistant. So DNase sensitive and a DNase resistant region. Same gene, different cell. So in this cell, the reason that it's sensitive is because it's loose, because that gene's expressed a lot. The reason that it's resistant to DNase is because it's tight. Why is it loose? Why is it tight? Maybe there's acetyl groups here. And we'll come back to that in a minute. But we haven't shown that. All we've shown is that, so what we know now is that it's expressed, because I kind of gave you that it's highly expressed. And now we've shown that the gene, the globin gene, is associated with loose chromatin in those cells, and the globin gene is associated with tight chromatin in those cells. This is just a picture of heterochromatin and euchromatin. You've heard those terms before. And this is just showing just a picture of it, that you can actually see heterochromatin stains differently in the nucleus. This is like an interphase nucleus. This is the nucleolus. This is just showing that heterochromatin actually means chromatin that stains differently. So there's something different about these dark spots. And what we know about that is that now the euchromatin is loose and heterochromatin is tight. It's more condensed. So it's kind of like an extreme example of this chromatin condensation stuff. And it shows that this is common in, so these marks are common in, so we know that heterochromatin is condensed, that the genes are off, and that chromatin is associated with specific methylation marks, whereas euchromatin, when the genes are on, are typically expressed with acetylation marks. There are some methylation marks associated with active chromatin, but for the most part, acetyl groups associated with open chromatin, methyl groups associated with closed chromatin. Now this isn't all that important. This is just showing that what happens. So here's some chromatin that's open. But after you methylate a specific histone, it causes it to get condensed. And there's this other protein called, so this is just showing you how heterochromatin forms. Heterochromatin protein 1. And I actually work with the lady who discovered that. I'm going to have dinner with her in a couple weeks in Orlando when we go to this genetics meeting. So this is the enzymes that add methyl groups are called methyltransferases. We'll come back to that in a minute. So this is not that important. It's just showing how chromatin is being associated, how chromatin is being formed, heterochromatin is being formed. Okay, so now, so here's what we know. We know so far that if a gene's on, well, we'll start with it being off. Gene's off, gene on. How would we know that? Yeah, we could do a northern blot. Northern blots are old-fashioned, but, you know, we'll just, we just somehow measure gene expression. We know that chromatin is tight. Say condensed, use the official word. Decondensed. Okay, so this looks like this. This looks like this. How do we know? DNA sensitivity. This is sensitive. This is resistant. So this is sensitive to DNase. This is resistant to DNase. So now we want to know what chemical marks are associated with these things. And I can tell you the answer, and when I've said this already, is that acetylation causes this to happen, and methylation causes this to happen. The enzymes that add methyl groups are called histone methyl transferases, or HMTs, because they're transferring a methyl group to histones. And these are histone acetyl transferases. So one more time, I need a little more room. Get more abstract as we go, tight. Loose. Histone acetyltransferases, histone deacetylases. So these have acetyl groups on them. The enzyme that adds those acetyl groups are histone acetyltransferases. The things that take them off are histone deacetylases. So we'll move this down a little bit. We also know that this is methylated by histone methyl transferase, and there's probably some demethylase. All right, so we're going to focus on this story. So the hats and the hat knots. No, the hats and the H-tags. All right, so how do we know that? So how do we know that loose chromatin has acetyl groups and tight chromatin has no acetyl groups? Let's do an experiment. So how do we know? It's like an after-school special. So now we have to talk about this other type of experiment, and I think this is pretty much the last one. It's called CHIP, for chromatin immunoprecipitation. Chapter 17 covers it a little bit. We've already covered this, but we're going to do it again. So this is basically it. Don't make it more complicated than it is. The key is, what are we going to isolate? We're interested in what's going on with chromatin, so let's isolate chromatin. So that means we're isolating chromatin, which is DNA and the histones associated with it. Then we're going to break it up into smaller bits. So we isolate it, and then we're going to precipitate it with an antibody. So if there's an acetyl group on here, we have an antibody that binds onto that. And then we can precipitate that in a test tube. So we got our antibody, and it's bound to our acetylated chromatin because this antibody is specific for this acetyl. It's specific for, you can make it specific for H3 lysine acetylate. I mean, it's very specific. And then that lysine is associated with a particular nucleosome, and then associated with that is a particular DNA sequence. All right, so we get the antibody that's bringing down the acetyl group, which is attached to the nucleosome, which brings down the DNA. So it's going to bring down a bunch of DNA. There's going to be a lot of different genes that are associated with acetylated histones, right? Lots of it. I mean, you got a particular cell, there's probably 10,000 genes that are being expressed. And all of those will probably be precipitated down into the bottom of this tube. But we just want to know if our gene is down there. Let's say that we're still interested in this globin gene. We just want to know, did our globin gene come down with this? If it did, then we can analyze it by PCR. So PCR is a technique where you can amplify up any specific sequence. So you have to have primers that stick to your globin gene and amplify that DNA and then run it on a gel. And if that gel shows that there's a band, then that gene was associated with this type of chromatin. If there's no band, then it wasn't there. Right? And then so what we'll see if we do this, so let's just do this experiment. let's take our erythroblasts. So we got our erythroblasts and our MSBs. And we're going to isolate chromatin, precipitate with an acetylated so let's say an let's say we're going to precipitate with an H3K9 acetylation antibody. Boy, there's a lot of abbreviations, but just an antibody that, so here this is loose. We expect that particular lysine to be acetylated. We've got so we break this up a little bit. We've got these acetyl groups. We precipitate these into the bottom of the tube using an antibody. And then we got this in the bottom of a tube. So we got all this stuff down there. We got our green stuff, got our red stuff, got our blue stuff. And associated with that blue stuff is some DNA. And so we do PCR and we run a gel. And in this band, in this lane, there will be, let's see, if we do an ethidium bromide staining, it will kind of look like that. So we'll have DNA in that lane, because this is from our EB cells. And we expect the DNA from our EB cells to be associated with acetylated histones. So here, that was our hypothesis. So we just did an experiment and we showed that yes, the DNA in the erythroblast was associated with acetylated histone. In the MSB cell, it's like this. The histones are not acetylated. We can still break it down. We can still add the antibody, but it's not going to precipitate, so we get no band. So that's no band. So in MSB cells, it's not associated with that. But what if we looked at methyl groups? Say we had some kind of methyl group on there. And if we use an antibody against the methyl group, then maybe the band would show up in the MSB cells, but not the EB cells. All right, so that's called a chromatin immunoprecipitation experiment, CHIP. And in the end, we use PCR. What we can also do is, because we're going to get a lot of DNA, so we're just looking for this specific gene. What if we want to expand this and say, okay, well, tell me all the DNA. So in this tube, there's going to be all kinds of DNA that precipitated out. So then we can just sequence it all, and that's called ChIP-seq, where we do the chip and then we sequence it. Now you can also do a chip and then do a microarray, and because microarrays are done on chips, that's called chip-chip. We're mainly going to talk about this for simplicity, and this when we expand it up to genomics. But it's fun to say chip-chip, so you got to throw that in there. So this is how the chip works. You isolate chromatin and break open the chromatin into smaller bits. Add an antibody specific for this acetyl group. Precipitate it. Isolate your DNA. So here's our DNA. Now we can do PCR or whatever to analyze what DNA is there. So when I say PCR, that means I want to know maybe there's a gene right here, and we have primers that flank that gene that allow us to do PCR. And if the band is there, that means that DNA has precipitated by that antibody. Close up of all that stuff. The reason it's called immuno is because whenever you use an antibody for anything, antibodies are part of the immune system, so that gives it the immuno thing. So you can see like a Western blot uses antibodies, it's also called an immunoblot. Anytime you see something like that, the word immuno in a technique, that just means there's an antibody involved. All right, don't worry about it. This is just showing, might as well show it. So we've talked so much about activation. We talked about like UASs, and a UAS can be bound by an activator protein, or we talked about an enhancer bound by an activator protein. This is just showing that sometimes you can have a repressor, and the repressor will have a DNA binding domain, a repression domain, and then these are co-repressors that will turn a gene off. And in this case, what they're doing is they're taking off acyl groups. And so that's going to lead to scrunched up chromatin. So it's just sort of the opposite of activation. This is one mechanism by which you can repress a gene in eukaryotes. And you can have an activator that binds the enhancer and adds acetyl groups. So that's another way. So you can actually have genes that are off. And it's a little bit counterintuitive because we're saying that if the gene is off, that you don't have access to this DNA. But apparently, I don't know what the details are, but somehow some proteins can get in there and bind onto this DNA, and then it causes this to be deacetylated, or acetylated in this case. So we're going to add acetyl groups. And this protein here happens to be a HAT, which is a histone acetyltransferase. Then this is just kind of rehashing this idea of the histone code, postulated by C. David Alice. And this is sort of the idea behind epigenetics. Chapter 21 goes into epigenetics, and I'll just leave that to you to read and do some of the questions in there. But now you know the basics. This is more than the basics. This is pretty detailed. All right. I think that might be it. Oh, we got one more slide. If I can get to it. All right, so that brings us back to this. So you should now understand what this means. This would be a good question. Explain this in as much detail as you can. So I'm just going to leave it at that and see if we can't shorten up this particular